Yeah. Yeah.
Okay, now I'll get started. So the title of the presentation is identifying non-mutational P53 loss of function in human cancers. So first, I'll give some background. Let's move this away. So we know there are many different mechanisms to regulate gene expression in cancers. We know the common mechanism is genetic mutation through somatic mutation or copy number deletion or structural variation. So some genes, particularly the tumor suppressor genes, will get reduced function, and oncogenes get activated function. So besides this, the somatic mutations, there are other non-genetic mechanisms that can also change gene expression. So this includes many different mechanisms, including the iron level and protein level. For example, alternative splicing, the non-coding iron regulation, epigenetic demethylation, and protein degradation. So there are so many non-genetic mechanisms that can change gene expression, but this mechanism cannot be easily detected through the DNA sequencing. So this is how to predict. The goal of this project is to predict P53 function. So how do we do that? We know P53 is a transcription factor, so we can find that P53 is the downstream target genes. So our hypothesis is if P53 function was changed, so the downstream gene expression will change also. On the other hand, from the downstream target genes gene expression, we can predict the upstream regulator, predict P53's status. So why we select P53? The first P53 is a tumor suppressor and is very critical in the cell cycle apoptosis and DNA damage repair. And also P53 is one of the most extensively studied genes in the literature, and the target genes of P53 have been well defined. And many of them actually are validated experimentally in either cell line or mouse models. Another interesting observation is P53 is the most frequently mutated gene in many different cancer types. I will show you later. So in general, the mutation frequency is over 40% in more than 10 different TCGA cancer types. And another thing I will show that another thing is we have a lot of cases carry the P53 truncating mutation. So truncating mutation makes the protein incomplete. So that totally loss of function. This can be used as training data in our model. So in terms of clinical significance, P53 mutation have been linked to the radiation or chemotherapy resistance. And also, because P53 is involved in the DNA damage repair, so P53 status is associated with the tumor mutation burden. And the tumor mutation burden is currently a marker for the immune therapy. So this is a table I calculated for using the TCGA data. You can see P53 mutation frequency is number one, is the top. You see the third column is the rank of the mutation. So it's the number one on dozens of different tumor types. So in this talk, I will mainly focus on two cancer types. We will try to predict the P53 mutation status in lung cancer and breast cancer. We select these two cancer types because several reasons. First, these two cancer types have the largest number of cases. Both of them have the over 1,000 cases. And second, and most importantly, I show in here, so you see they have a large number of P53 truncating mutations. And this will be used as a training data in our machine learning models. basically we use normal sample and truncating sample as the training data. So we assume P53 function in the normal samples are in normal condition, and P53 complete loss function in the truncating mutation cases. And the same thing for the lung cancer. And after we train the model, we will use as a VA model to predict the other classes, predict the missense mutation cases and the wild-type cases. So this is general workflow of our work. First, we see from this panel. First, we identify the P53 target from the literature, and we collect all the reported P53 target. And then we further validate this target using the P53 ChIP-seq data. So we assume if a real P53 target must have a P53 binding at its promoter. Then we also analyze the GTX expression data to make sure all these target genes have reasonably expression level in different tissues. We want the target genes have a relative high abundance. And then we use this, as I said, I use a normal sample and the truncating mutation sample of the training data to make a SVM model. After we build a model, validation model, then we can use the model to predict the P53 wild type and P53 missense cases. So this is to give you an example of how we about the P53 target genes we identified. So we can see in this, we use the published data. This is actually a breast cancer cell line data which have P53 wild type MCF7. So these genes are P53, activated by P53. And you can see here is the control. The P53 means P53 is inactivated because in normal condition, P53 have very low activity. However, if the cell was treated with the gamma radiation, P53 will get activated because gamma radiation induces DNA damage, and P53 become activated to in response to DNA damage. You can see after P53 get activated, you see a significant increased gene expression. And the same thing for this target gene called BTG2. So in 0 hour, which means P53 is inactivated, you see no binding, almost no binding. But after gamma radiation for 0 hour, you see obvious clear binding of P53. So this just to give you an example of the P53 target gene, how they respond to the cellular stress like the gamma radiation. So result. So these two results, as I said, I only show the lung cancer and breast cancer. They are same type of data. So I focus on the left is the lung cancer. So in lung cancer, for example, we identify 23 genes that are activated by P53 here. We also identify the 32 genes that are repressed by P53. So after we divide all the lung cancer patients into these 4 groups: normal, wild type, missense, or infrared mutation, and truncating mutation. So you can see the P53 mutation expression levels are changed, are different. So especially if you see 2 big pattern here. So in the missense and truncating mutation, you see P53 activated genes are low expressed in these 2 classes, but high in normal. However, for the repressed genes, you see the opposite trend. So the expression is low in the normal condition and is high in the P53 truncating and missense mutation. The interesting thing is the wild type. So you see the wild type cases, the expression pattern is staying in between. So this suggests many of the wild type samples actually have the P53 loss of function, even though you don't see genetic mutation, but the P53 protein indeed have lost some function. We will validate this later. So the same pattern for the breast cancer. We also did this for other cancer types, but did not put the data here. So then for each cancer type, we'll have a list of P53 target genes. we don't want to use all these genes. We instead of use each genes as a feature to train the model, we calculated a composite expression score. Composite expression score means you use a single score to measure the expression of a group of genes. So this score, measures genes are coordinately up or down regulated within a sample. So one sample, one score. And we use 4 different algorithms. All these algorithms are published or were established to calculate the CES. We use the GSVA, SSGSVA, and PCA. We use the first principal component and the Z-score. So why we use the CES rather than the single gene expression? Because the CES can provide intuitive and stable measurement. If you look at only one gene or a few genes, they may change. They have variability, and it also reduces number of features, which can avoid model overfitting in our cases. It also reduces noise, technical, and biological variabilities because the group of gene expression is relatively stable. And it's also possible to do the pan-cancer analysis. So this I show the composite expression score in the using the P53 activated gene and P53 repressed gene. So the up panel showing the CES score calculated from P53 activated genes. The bottom panel showing P53 CES score calculated from P53 repressed genes. So similar before, we divided the samples into 4 groups: normal and tumor. Tumor, we have 3 subgroups: P53 wild type, P53 missense or infrared, and P53 truncating mutation. So you can see clearly in normal condition, P53 active genes are high but low in the P53 mutation cases. And you see the opposite trend for P53 repressed genes. So the same thing if you use different algorithm, use SSGSEA or this score. Yeah. So you can see in this slide, if you compare the different score, we compare the score calculated from P53 activated genes and P53 repressed genes, you'll see a very good negative correlation. And it's the same thing for the other different algorithms. So this suggests these activated genes or repressed genes are indeed show the opposite trend. So this is the SVM model we are trying to build in this project to predict. So first, the goal of this model is to predict if the P53 protein function is normal or loss of function. And we use SVM model because this is a binary classification. So our prediction is basically yes or no. If you give the model a list of the gene expression, it will tell you if the P53 is loss of function or not. So with the linear classification, and we have 7 features for the CES score, because for the first 3 features we have both activated score and repressed score. For the principal component analysis, only the PC1. So in total, we have 7 features. And we demonstrated the SVM model in 2 kind of types, lung cancer and breast cancer modeling. The training sample, I said, we use normal and the truncating mutation cases for the training. So this actually, we have 7 different features. We visualize 3 of them. So we visualize the principal component, GSV, SS, GSE. So and then we color the patient using different color based on the P53 status. For example, this color represent normal samples. The remaining are tumor samples. In tumor samples, you see the orange are wild type, red are missense, and purple are truncating mutation. So you can see in the 3D space, all these samples are indeed kind of separate very well. So in terms of the model, the SVM will try to find a hyperplane to separate these samples. And you get the same thing. You find a very similar conclusion for the CES score calculated from repressed genes, just sort of the trend. So this is the ROC curve showing the performance of the model after five-fold cross-validation. Five-fold basically means we for lung cancer, for example, we take 80% sample of the training data and use the remaining 20% sample to validate the model. So we did this 5 times. This is called a five-fold cross-validation, and we show the prediction accuracy. We use the F1 score, precision recall. All these scores, have excellent value. So this table gives you the overall performance score for our model in different cancer types. We did lung, breast, colon cancer, esophagus, and also we did this analysis in the pan cancer. In the pan cancer analysis, we combined 12 different cancer types. So then we use after we build and validate the SVM model, we use the SVM model to predict the other cases. In this case, we use the SVM model to predict the wild type and the missense mutation cases. As shown in this sunburst plot, these are training data, and this is a wild type, and this is a missense. And the out panel showing the prediction result. So you can see in the wild type cases, these cases all have the P53 wild type, so no mutation. And you can see 94% out of 330 cases, 310 are predicted to be loss of function. Only 6% are predicted to be normal. For the missense mutation, 100% are predicted to be loss of function. So then we compare these cases. We compare the overall survival of this lung cancer patient. What's the difference between this predict normal and predict loss of function? As shown in this panel, you can see for the patient that are predicted to be normal, they have a much better overall survival compared to the patient that are predicted to be loss of function, which is the orange curve. So in another word, for those cases that are predicted to be loss of function, even though they're genetically wild type, they have the same, almost the same overall survival as the missense and truncated mutation. And this is your Cox regression to show the patient predicted normal indeed have a much better. For example, if we use this model to predict lung cancer survival, you can see at 10 years, 112 months, the other cases patients have P53 mutation, the survival rate is only 25%, but is 75% for the cases that are not predicted to be normal. However, if you combine this pretty normal and pretty loss of function, you cannot see this trend because this case is much smaller. So we can validate our prediction from another angle. So the upper panel showing the original grouping, we did not predict the P53 status. The bottom panel showing after we divided the wild type patient into two groups, pretty normal and pretty low function. So in the first column, we see the tumor mutation burden. So the Y value, YX indicate the tumor mutation burden. You can see before prediction, if you compare the P53 wild type to P53 missense mutation or P53 truncated mutation, you can see they are a little bit different. The wild type cases indeed have a lower mutation. However, if we use the SVM model to predict, to divide this group into two subgroups, so this is a pretty normal and a pretty low function. You can see the cases that are predicted to be normal have a much lower tumor burden compared to the cases that are predicted to be loss of function. And we see the same trend for the other measurement. For example, the fraction of genome alter. This is also called the CNV burden, copy number variation burden. So we see the similar trend. And annual product score is another type of copy number. It's a chromosome, whole chromosome level, whole genome level duplication called aneuploidy. So you will see the same trend. The predict cases pretty normal have a much lower score. So this is the hypothesis score, because I'm not talking about too much about P53 function here, because P53 also involved in the hypothesis signaling pathway. So you can see after prediction, we indeed can separate the wild-type patient into two separate groups, and they exhibit completely different genomic characteristics. So as I said, as we see previously, many of the majority of the wild-type cases are predicted to be loss of function. So we term this sample of P53 wild-type predict loss of function. So why they have so many samples predicted? So we want to dig into this issue. First, we found that 12% of this P53 wild-type are actually have P53 mutation, but for some reason they were missed by the TCGA variant code. So this gives you an example. We detect this mutation from the RNA-seq data, but this cases was missed by the TCGA. You can obviously see the mutant allele fraction is 45%. In this case is this C indicated to A, and we found 12 samples. So 12% samples have these cases, and another 28% of these wild-type patients actually have the MDM2 and MDM4 amplification. So if you know P53, these two proteins are the P53 upstream regulator. It actually repress P53 function. So if this gene get amplified, get activated, then the P53 protein function will significantly repressed. But in addition to this 12% and 20, there are remaining 60%. We don't know. Maybe for other epigenetic reasons, this P53 loss of function. So the conclusion is the non-mutational P53 loss of function, or we call non-mutational P53 gene activation is prevalent. Actually, the majority of the wild-type patient have this P53 gene activation, and these samples, those wild-type samples that are predicted to be loss of function, have different genomic and clinical characteristics, including tumor burden, CNV burden, high hypoxia, and significantly reduced overall survival. So this P53 wild-type patient predicted to normal have the best survival, have the best prognosis, and we found a total of 40% of patients P53 wild-type patient have the loss of P53 loss of function can be explained by either the false negative mutation code or the MDM2 and MDM4 amplification. So some of the work are done by two students in the Beijing Genomic Institute in China, and some of the work are doing in Mayo Clinic. So I'll take any questions if you have.